There is a worm that's called ice worm that's a marine polyp that lives deep in ocean trenches.
Did actually know.
But that's not the ice worm we're going to be talking about. Tristenne grew up in Denver, went to the School of the Arts there, played piano, and came here because there's both marine bio and a good music program. And so she's majoring in bio, minoring in piano performance, and doing the honors program. When she is not a student, she's worked for most of her time here, including in an escape room. So if you want someone who knows escape rooms, take Tristenne with you. And also in a rage room. And while she is not doing any of those things, she sometimes knits ice worm patterns. And ice worm patterns. Clothing. So, with all that, Tristenne, tell us about metabolic rates and ice worms. It's been so fun to work with.
Okay, well, hi. If you guys don't know already, my name is Tristenne Cranford. And today I will be telling you. The projector doesn't. Okay, well, we'll figure that out. My presentation is on examining the effect of temperature on metabolic rate in ice worms. Their scientific name is Mesenchitreus solificus. And I'm gonna pull in a little bit of like English honors sort of here. Solificus essentially means sun avoiding. So there's a little sneak peek on ice worms. But first, and this is a last minute addition into my presentation, ice worms were featured in the newspaper The Week. There's this little snippet on them, but most importantly, Peter and I saw this last sentence, which says, most organisms slow their metabolism as they get cold, but ice worms produce more energy the colder it gets, which we thought was a very interesting statement, and we'll see why through this presentation. So that's my little hook. We were in a newspaper, and I have questions about So a little bit on ice worms. They live their entire life in the ice and snow. This scale bar here is in millimeters, so they're about 20 millimeters long at their longest, so pretty small. And they are actually the largest animal that lives their entire life in the ice and snow. They're born in the snow, they reproduce in the snow, they die in the snow. So pretty small creature to be the largest creature to do something. They eat bacteria and algae. So in the summer, they come up to the surface of the snow and they eat all the little microbes and bacteria at the surface. And then at night, when that top layer freezes, they actually burrow down into the snow because these glaciers are 0 degrees all the way through, which leads me to their range. This really dark pink, and I'm so sorry, Katie Jane, I know you're colorblind, but I didn't have another way to do it. They range from southern Alaska to central Oregon. So they live on glaciers all along the coast of North America here. And they live on what are called temperate glaciers, which sounds like an oxymoron. But these glaciers are only 0 degrees Celsius all the way through, so they don't really get below freezing, which is why ice worms can burrow into the snow, into that insulating layer, and not freeze because they are insulated by all of the snow. So how do ice worms live in these environments? They live at about 0 degrees Celsius for their entire lives, which for those of you who aren't super familiar with Celsius is freezing, 32 degrees Fahrenheit. And they are extremophiles. They live their entire life at freezing, which is, I would say, a pretty extreme temperature. And this means that they are very vulnerable. They're very vulnerable to heat. In climate change, our modern era, they're very vulnerable to overheating. And it's really important to study ice worms because they are extremophiles who can't tolerate freezing. If they freeze, they die, which is very absurd because they live their entire lives at freezing. So they are very vulnerable, and it's really important to study them because if we study how they have adapted to the cold without adapting to freezing, then we can move that research onto creatures that live in maybe a little more habitable environments, and we can say, oh, we have this extreme, maybe there's similarities in the way other animals adapt to the cold. Speaking of other things that adapt to the cold, we have a couple different ways we've seen animals adapt. This Antarctic fish has evolved an antifreeze protein, which essentially disallows the liquids in the key parts of their body from freezing. So they make their liquids in their body have a freezing point that is lower than the freezing point of seawater. So unless the entire ocean froze, they would not freeze themselves. Similarly, there's this frog that lives in the Himalayas, which ups the solute concentrations in its body, which also lowers the freezing point of all of their fluids and prevents them from freezing because their freezing point is actually lower than freezing. Ice worms, as far as we know, don't have these. They don't have antifreeze proteins, but some studies have been done that do suggest that they adapt by increasing their metabolic rates. Hold on, you say, I'm an English major, so what is a metabolic rate? I'm here to let you know. The metabolic rate is the rate of energy production within the body. I will refer to it sometimes in this presentation as MR, so if you're taking notes, MR equals metabolic rate, just so you know. And the way that you can think about this is you eat your carbs, your protein, your fat, your hamburgers, your grilled cheese, all that good stuff. And if there is oxygen around, which for you, hopefully there is, you create ATP, which is essentially an energy currency within the body. And then this ATP can be used if you're burning it, if you're going for a jog, it will increase your metabolic rate. So your breathing increases, your heart rate increases, and that means that your body is producing more ATP so that you can run and your metabolic rate increases. So that's a little spark notes on metabolic rate. But your metabolic rate is different from what a cold-blooded creature's metabolic rate would look like in the cold. So when warm-blooded creatures get cold, like this little mouse here, they increase their metabolic rate so that they can stay warm. So if you think about when you get cold, you shiver, and that is your metabolic rate going up so that you can stay warm. Cold-blooded creatures, however, their metabolic rates decrease just due to the chemical reactions in their body slowing. So if you think about dissolving salt in a pot of water, it's a lot easier for that salt to dissolve if the water is hot than if the water is cold. So essentially, the chemical reactions in cold-blooded creatures are slowing, and they're not doing anything to increase their metabolic rate. So as temperatures go down, their metabolic rate also goes down, but also as temperatures increase, their metabolic rate goes up until they reach a point where they can't perform as well anymore. They reach their limit of life, and their metabolic rate goes down because they're dying. So that's not good. But in both of these cases, whether either of these creatures get way too cold or either of these creatures get way too hot, extremes kill. Extremes are very bad for most creatures. So a little background info on ice worms and their relatives. This little white guy here is a potworm. They're a very close relative of ice worms, and they can tolerate freezing temperatures. So they are a tundra species that are cold adapted. They've been found in Germany, Svalbard, if you know where that is. And they can survive temperatures up to -5 degrees Celsius, which is below freezing. Ice worms, we found, cannot survive at these temperatures. They freeze and they die. We believe that these potworms can survive up to -5 degrees Celsius and warm up and be pretty good because they have accumulated carbohydrates in their body. So if you think about the frog and the fish that I showed you earlier, they have a higher solute concentration in their body, which then lowers the freezing point of the key structures in their body. So then key things that they need to survive don't freeze within their body. Specifically in ice worms, studies have been done that show that ATP increases as temperatures decrease. And if you think about this little graphic that I showed you earlier, if ATP is increasing, that means they have more ATP to burn to increase their metabolic rate. And in addition, mitochondrial inorganic polyphosphates, which are just another energy currency, also increase as temperatures decrease. So this is really interesting given what I just told you about metabolic rates. I told you that metabolic rates of cold-blooded creatures go down in the cold. So why is there ATP going up in the cold? That doesn't make any sense. This is a graph pulled directly from the paper that I just mentioned, where ATP is high when temperatures are low, and then it's dropping as temperatures are rising. So there's a couple, I'll go back. We believe, we don't know why this is. Other species, metabolic rates ATP did drop as temperatures went down. So ice worms reacted opposite from other temperate creatures. So this is really interesting, but we realized that nobody has made a whole body metabolic rate graph of ice worms before. Nobody has made a graph like this of ice worms. So we were thinking maybe ATP goes up as temperatures go down. So what if their metabolic rate goes up as temperatures go down as well? So just a little bit about whole body metabolic rate before I go into my methods. It's most often measured with respirometry. So they measure oxygen consumption of a creature. So this is a little fish in a container. They've locked him in there, and they are consuming oxygen, and the scientists are measuring the consumption of that oxygen. So this is how I measured whole body metabolic rate in the ice worms. So I had a couple of questions that go under this big sub-question: how does temperature affect the whole body metabolic rate of ice worms? So a couple sub-questions. Do these results provide insight into the ice worm's limits to life? Like I showed you before, this is sort of the limit to life where their metabolic rate can't go any higher, drops off, and they start to die. So can I get the ice worms to that point where I can find out their sort of highest temperature that they can survive? Are these studies consistent with the studies that were done with ATP? And how do their metabolic rates compare to potworms, which are their relative that I showed you earlier? So now I get to go into the super fun stuff of what I actually did. So I went with Peter Wimberger and Maggie Smith, Hannah Robideau, and Evie Stantart, and we all went up Mount Rainier. It was a super beautiful hike. This is me before. This is all of us about midway up. It was pretty cold, even though it was June. It was summertime. Snow was covering the ground still. And this is my sort of after. We're at the top of Mount Rainier with those ice worms. It was only a 3-mile hike, but it was a 3,000-foot gain in elevation. So super fun. Did it once. Maybe would do it again, but you might have to pay me some money to do it after. Peter was a trooper. He had a torn ACL at the time, and he was, oh my gosh, amazing. But we went up there and collected these ice worms with the snow, put them in little baggies, and then we came back down. Down was much more fun. We got to slide down these little snow chutes. Took us about a third of the time to go down as it did to go up, so that was pretty fun. But collecting these worms was actually the easiest part of the hike, because this is what it looks like at the top. This is about 3,000 meters in elevation, which is about 8,000 feet. And they just litter the snow. My tactic was to sit in one spot with my little tweezers and just put them in the bag, and I got more than 300 worms that way without moving. So it's about 100 worms per cubic foot of snow. So there's no mixing them. They're all over the place. This is, I will say, during the summer, in the middle of the day. So this is when that top layer is nice and warm. They're eating all the bacteria and such. So this was the perfect time to go up, and Peter planned it as such. After I had gotten the ice worms, I then drove all the way to Montana, and I worked with Dr. Rachel Mallison and all of her interns at Flathead Lake Biological Station. It's a beautiful station, and Dr. Rachel Mallison taught me a lot about respirometry procedures. So She helped me with my ice worms, and so did her interns, and she works on stoneflies. So I also got to help her collect stoneflies. It was very fun. So we deemed ourselves Team Icefly. It was a really, really fun time. And for each trial that we did, we put 12, I'm gonna have a little influencer moment. We put 12 worms into these little tiny bags. I sewed these myself. It was, I wish I had a magnifying glass, but they were very fun to sew. And we put 12 worms in each bag. So this is 1 worm in a bag, and that is 12 worms in a bag, all closed up and ready to go into the equipment. We had to be very gentle with them, putting them into the bags, because if you roughed them up a little too much, they would fall apart. And that's no good. They don't breathe if they're dead. So I couldn't measure their oxygen. So we had to take a little dropper and squirt water into the bags, because the bags were mesh. So we kind of washed them into the bottom of the bags. And every once in a while, we achieved what we call a one squirt wonder. So you squirt it into the bag, and that was all you needed to do, and it was amazing, which is where that Snapchat came from. After they were put in the bags, we put them into these chambers. So this glass is frosted, unfortunately, but you can kind of see the little outline of the bag here. There are worms in there, and they're all sealed up, and they're breathing so that we can measure them. We measured them at temperatures between 4 degrees Celsius and 22 degrees Celsius. So for our Fahrenheit people, 4 degrees Celsius is pretty close to freezing, so pretty cold. But 22 degrees Celsius is not this room. This room's a little warmer, but 22 degrees Celsius is about room temperature, probably closer to the temperature in the hallway. It's hot in here. And then before we put them into the bags, we weighed them, and we wrote down that data so that mass-specific metabolic rates could be calculated. Now, don't let this scare you. It just means that it's the metabolic rate per gram of animal. So that way we can standardize the metabolic rate if one worm is bigger than another. And these worms are really, really small. They weigh 0.7 milligrams each about, so really, really tiny little critters. So once we had them in the bags, we got to put them in this lovely equipment that Dr. Rachel Mallison let me use. So it all starts with this water cooler here. We set it to whatever temperature we want the water to be at. And it goes through, I have the yardstick. It goes through this metal coil that is in this tub here. And then this coil cools the water that is inside this tub. The water inside this tub goes through these tubes into all these clear boxes. So each box has 4 spots for chambers in it. So we could have 8 chambers going at a time, which is really, really lovely. And this is me closing up a chamber for the worms. So this is the chamber. Here's the worms. This is the end of the chamber where this little tube is, and then the worms are in between my hands here. So we can do a couple replicates at a time with this equipment. And all these little black cords that you see around here are fiber optic cables, which could then sit on the top of the chamber, and it would measure the oxygen levels within the chamber. So that was how we measured how much oxygen they were breathing, because as they respired, the oxygen levels would go down in the chamber. So this is kind of what we expected a graph to look like coming straight out of those chambers. We measured the oxygen levels with a very fancy software that Rachel had called AutoResp, which is through Low Ecosystems, and we left one of those 8 chambers empty. So we did that to control for any microbes that were respiring in there, any maybe leakage that the tubes had because they were just sealed with frosted glass, and we left them in there until the oxygen was fully depleted. Excuse me. So that could have taken anywhere from 8 to 18 hours, depending on the temperature that they were run at. And then this was used to calculate metabolic rate later. So this is our example graph. This is a control chamber that is slowly dropping in oxygen levels because microbes are respiring or there's a little tiny leakage, whatever that may be. And then this red line is a chamber with worms in it. So you can see the chambers are breathing and they're depleting that oxygen. And then here, they don't have enough oxygen to properly run their body anymore. So they reach what we call a break point, where they essentially pass out, you can think of it that way, and then they consume a lot less oxygen over a greater amount of time. So this break point will be very important, so write it in your notes. And then this is the graph we actually got. Very exciting. This is a graph of one trial at one temperature. So this is the 7 chambers that actually had worms in it, and this is the chamber that had no worms in it. And you can see here that there was a break point where these worms eventually passed out. And it's not surprising that all of these lines are really different, because worms weigh different amounts, because they're creatures and they can't all be exactly the same. So some of the larger worms would consume more oxygen quicker than the smaller worms. But this is standardized for, because remember, I weighed them, so that's going to come in. So before I get into these results here, I'm going to ground you within what this graph means. So this orange line is the hotter worms, and the blue line is the colder worms. See what I did there? Sorry, Katie. And, this 4-degree line correlates to this 4-degree point right here. And then this 16-degree line correlates to kind of over here. It would be this point, but it would be this point on the line in the graph. So you can see here that the slope of this line is going down a lot slower than the slope of this line. They were consuming oxygen a lot less quickly at 4 degrees than they were at 16 degrees Celsius. So because they were consuming oxygen slower here, their metabolic rate is lower here, and they consumed oxygen really, really fast at 16 degrees Celsius, and so their metabolic rate is higher here. So that's to sort of ground you in this graph here. So we did find that there was a significant increase in metabolic rate from 4 degrees to 16 degrees here. And we did kind of find their limit of life. If you think back to that line that I showed you earlier that had the little beetle on it, that's kind of what this line looks like, which is very, very exciting. We found that their limit of life is about 14.5 degrees Celsius. However, all of the worms that I had put in the chambers had essentially disintegrated at any temperature above 7 degrees Celsius. But they were left in there for, 10, 15 hours, something like that. So I do have, at the end, I have the worms that stayed intact at 4 degrees to 7 degrees Celsius. So if you want to come up and look at those later, you are absolutely welcome to. But it is interesting that their limit of life is around 14.5 degrees Celsius. But when I took them out of the chambers, they were all disintegrated at any temperature above 7 degrees Celsius. So this is important. Remember this for later. I'm also going to ground you in these breakpoint results. So you can see the breakpoint here, where they are consuming a lot less oxygen, and breakpoint kind of here, where they are also consuming a lot less oxygen. So at 4 degrees Celsius, they passed out after a little over 8 hours, and so you can see this point here correlates to hours there. And then over here, the worms passed out a little before 2 hours in the machine, and you can see that point down here correlates with the hours over here. So what we found from this results was essentially that the warmer the temperature was, the shorter time it took for the worms to pass out, which is expected for cold-blooded animals because they change their metabolic rates with temperature, because they don't make an effort to change them. So to go back to the questions that I asked at the very beginning, does this provide insight into the ice worm's limit to life? Yes, we found that it was 14.5 degrees Celsius, but more research does need to be done. If you also remember, I told you that the worms disintegrated at about 7 degrees Celsius, which doesn't make sense, but actually it does make sense, because if you remember, you need oxygen to produce ATP, and I had locked these ice worms in little tubes where they were consuming all the oxygen, and then they didn't have oxygen by the end. So they probably disintegrated at above 7 degrees Celsius because I didn't give them oxygen. So more research needs to be done to really know what their limit of life is, if they can be held at 14.5 degrees for long periods of time and still survive, or if that's just where their metabolic rate drops off, or if they do disintegrate above 7 degrees Celsius. But we're not quite sure because I didn't give them oxygen, which is important to life. How does temperature, how are these consistent with the ATP studies? So if you remember what I said before, they said their ATP went up as temperatures went down, but my metabolic rate was lower as temperatures went down. So they don't exactly correlate. However, it might make more sense that their ATP rises as temperatures drop because they're trying to prevent their metabolic rate from going lower than it actually is. So more research needs to be done, I think, with ATP in conjunction with the metabolic rate and perhaps with their relatives, the potworms, to see how the ATP is maybe helping them increase their metabolic rate or if it's for something else entirely. Speaking of the potworms, how does my research compare to the potworm research? So Sue and I pulled this data from another paper. This is from German potworms and Svalbard potworms, and they measured potworm metabolic rate with the production of carbon dioxide, which is different from what I did because I measured the consumption of oxygen. So if you remember, you breathe in oxygen, you breathe out carbon dioxide. So it's a little bit of a different method, so take this graph with a grain of salt. However, we do believe that this shows that ice worms are more cold adapted than potworms because their metabolic rate is higher than potworms at lower temperatures. But more research needs to be done. That is always the mantra of science, to do more research. And it's really important to understand why ice worms are more cold adapted than potworms and how they achieve this, because again, if we study extremophiles, we can then further better understand our more temperate creatures that we study. So that's my presentation. I would like to thank so many people, obviously Peter Wimberger and Sue Cranford, who have brought me in on this research and have been helping me write my paper and everything. Rachel Mallison, who I work with in Montana, Scott Hotaling, who is basically Mr. Ice Worm and has given me a lot of information on this. Obviously, everybody that I went up with to collect ice worms, Team Icefly, which are all the people that I worked with in Montana, and everybody who helped me there. Obviously, I couldn't do this without funding, so thank you to the McCormick grant. Obviously, the bio storeroom gave me a lot of ice and styrofoam boxes and things so that I can help the worms. I would like to thank Liam Kimball for letting me know that Peter Wimberger needed a summer research student. And I said, yeah, I'll do summer research. And so now I'm Ice Worm Girl. So and obviously, I'd like to thank the worms. I don't think we thank our study creatures enough. So thank you, Ice Worm. Are there any questions?
Do you know what temperature the ice worms do freeze at?
They do freeze at 0, I believe, is what. Just a little bit below 0.
Okay.
Because they do have a little bit of those solutes in their body, but not enough to prevent them from actually freezing it.
So between 0 and -1?
I would think so. Yeah, just a little below 0, because they're basically at 0 for their whole life.
You talked about in the beginning how this research could be significant for the study of other sort of creatures that live in extreme biomes or cold sensitive or heat sensitive creatures. Could you talk a little bit about, this is a general question, how that might be applied to maybe other studies?
Yeah, so if we figure out that ice worms adapt to the cold by doing X, and we see another creature who perhaps lives in a varying biome, like the desert where it's really cold at night but really hot at other times, we could find similarities in what the ice worms do and perhaps what a lizard does in the desert. Or even if there is an animal that lives at a decently normal temperature but can tolerate freezing in the winter, or not tolerate freezing, but can adapt to the cold in the winter. we could just see if there's similarities there. And so then we sort of have something to go back to say, hey, ice worms do this, and they can't freeze. So what is this animal doing? Yeah.
Super cool presentation.
Thank you.
So it surprised me how adapted they were to, 14 and a half degrees and they're still surviving. Do you get, is that surprising in and of itself? And then also, do you think that, the amount of time you give them to adapt to that temperature might influence how well they can sustain the temperature change?
Yeah, absolutely. So this was surprising to me. It probably wouldn't be surprising to Peter because he's left ice worms just like open in the lab and forgot about them. They've been alive when he got back. But that was surprising to me. Certainly if you give, like seasonally, if you gave ice worms enough time to adapt to that temperature, they would do a little better. And obviously there is limits to that. But cold-blooded creatures certainly seasonally have a cycle where in the cold, their metabolic rates go up to to account for the cold, and then once it gets warmer, that line will go down. So then they have a cycle. But yes, certainly they can acclimate.
So when you were talking a little bit about more research needing to be done, and you were saying that you would need to feed them more oxygen to see actually how long they'd be able to survive, how would you then measure their metabolic rate if you're continuously giving them oxygen? Is the carbon dioxide output then your measurement marker? If you understand my question. Okay.
That's a great question. Yeah, you could. It is hard because ice worms are an aquatic creature, and carbon dioxide dissolves really easily in water. So that's why the pot worms were measured with carbon dioxide, because they are not aquatic. They are terrestrial.
So you need something else that's not oxygen and not CO2 to measure.
Yeah.
Interesting.
Yeah, I don't know. Any thoughts on that? How you would well, you couldn't measure ATP at the same time as metabolic rate anyway, because you would have to grind them up. That's how they measured ATP, was they, I think, flash froze them and then ground them up, and then you measure their ATP that way. But
if you just want to know if they survive, you could lock them at a temperature with oxygen in there.
yeah.
Then you wouldn't be measuring metabolic rate, you just see if they disintegrate. So I think you could do that.
Yeah. So that would be for survival. But
I know it's hard for you because you're an escape room person, so the idea of
giving them
out is hard, yeah.
Oh. Terrible. Yeah. So that's okay. Yes, Amy.
When you say they disintegrate, you mean they actually disintegrate? They fall apart?
Like, yeah. So funny story. I got this in the mail when I was in Montana, and that's ice worms. So they really do. Their cells disintegrate and they turn into water, basically. Brown water. Yeah, so this was a mishap when I got ice worms mailed to me. This is 1,000 ice worms. Maggie and Evie had been so kind to hike up Mount Rainier again for me and mail to me, and so they obviously put ice in the package, and then instead of coming straight to me to Montana, it went to Tennessee, and then it came back to Montana, so then that's how I received those ice worms. So yes, they just turned into water. So these were the worms only that were collected at like 4 degrees Celsius, because they were the only ones that did not dissociate when I did my experiment. Because otherwise I just have like brown water. And, yeah.
And they have there's a lot of fats, a lot of liquids in them. And so like you put them in your hand and just let them sit. It's like M&Ms.
Yeah, sort of building on that, I guess I'm curious. It doesn't directly relate to your research, which is fascinating, but it's connected when you look at climate change and stuff like that. I'm kind of wondering, the life cycle of a worm. I don't know very well, but I know it involves eggs, and or at least a lot of worms I know it involves eggs. And the eggs might have a different, might respond differently to changing temperatures, such that maybe, you know, if we've got climate change, warming happening, and it's not it becomes a hostile environment to the worms, the eggs may linger long enough for, how long would they linger? Anyway, just, I'm wondering how that relates to the bigger question of worm survival over the course of climate change.
Funny, we also don't know a whole lot about their life history. That was another student's project, was to look at the life history of the ice worms and how they reproduce and when they reproduce, and if they live longer than a year to then reproduce in the next season. So I don't know a whole lot about how the sort of reproduction would change with higher temperatures, because we don't know a lot about how they reproduce at all. But that would be interesting to see if their sort of egg sac would be more vulnerable to higher temperatures, or if it would be less vulnerable. But that would be a really cool study.
Thank you.
Yeah, thanks.
You mentioned that ice worms are partners of bacteria, and I was just wondering how that might affect and or benefit other species that inhabit similar climates. If you want to talk about that?
Can you elaborate on your question? How do the worms then be beneficial to other species that
Yeah,
we
like
eat
are
them?
we consuming bacteria that would be harmful to other species? And so like with global warming, if their habitat is dissipating, like would that impact other species? I don't know how many species are like surviving in these types of climates. You said the ice worms are the only ones that live their whole lives. But
They're the only ones that live their whole lives in the snow. Well, they're the largest ones that live their whole lives in the snow. Oh no. The glaciers are actually more inhabited than you think. There are a lot of birds. That's right. A lot of birds that eat the worms, and from what's been observed, the ice worms probably don't eat harmful bacteria that would then harm any birds that would eat them. One of Scott Hotelling, who I've been working with and has been up there and observed sort of the ecosystem that they're part of, has seen that the ice worms don't really make an effort to run away from birds. He says the fastest that you'll ever see an ice worm move is if it's cold on the surface. They will burrow down so fast and you won't be able to find them. So less of their survival instinct is about being preyed on, and it's more about not freezing. That's an awesome question. Yeah, awesome.
I have a very curious question.
Yes.
So what would happen if someone were to hypothetically eat an ice worm?
Oh, Peter knows. I honestly wish I had eaten one.
You would turn into me.
You can't eat the ice worm and let
I can't eat them, and apparently they don't taste like much just because they're so small. I don't know what it would be like if you ate like a spoonful of ice cream. Oh yeah. Put it in my water in the morning, like an infused.
Can you research how many calories an ice worm has?
Yeah, how much protein.
This is fitting for honors program. There is a poem by the Alaskan poet Robert Sermon called "The Ballad of the Ice Worm Cocktail." I highly recommend it. It's pretty funny.
I didn't know that. Yeah. That would have been good study for my presentation, Peter.
We're trying to get that thesis written.
It's true. I could put a quote in there. Katie.
When you got your graphs with the breakpoints and such, how did you compare it to the control? Like, did you subtract the controls out, or did you?
Yes. Yeah. So I did some coding, and yes, subtracted essentially the slope of the control from the slope of the ice worm. So yeah, this would be a lot more sort of poignant without that control. So yeah, that's not. So did you have a question?
I was just gonna say, so you find them like on glaciers, or can you find them like in just like say the snow's falling, can you find them there, or is it specifically like at elevation and like?
It is specifically at elevation. There wouldn't be a whole lot of opportunity for them to travel to lower elevations and then show up when it snows. So it's only in the places where there is permanent ice and snow because it's required for them to live and reproduce.
They do have relatives that live at lower elevations in snow, and they go into sort of water and mud during the summer.
I have at least 7 more minutes.
Okay, I have an unrelated question. Does anyone know?
I actually don't. So if somebody knows what that balcony is for, I'd love to. I wonder that every time I come here.
Physics. Physics demonstration.
Oh.
Dropping things.
Big things in life.
Eggs.
Babies.
If there aren't any more questions, let's thank Tristenne.
If anybody wants to come see worms or the tiny little bag that I sewed, they are up here. But feel free. I'm sure there's still gummy worms outside, and so they can have a newfound appreciation for them. Yeah. Thank you all for coming. I really, really appreciate it. Thank you for not making me present to like 4 people. That's really awesome. Thank you guys.
Tristan, you should shout out Liam's thesis on Monday.
William Campbell is presenting his thesis in this exact same room on Monday at 4?
Same time, yeah.
At 4 o'clock. So you should come learn about electromagnets.
It's gonna be a lot of fun.
Yeah.